Jaekelopterus Rhenaniae was a giant aquatic creature that lived in ancient times, belonging to the group known as eurypterids. It was part of the chelicerates, which are related to spiders, scorpions, and other claw-bearing arthropods. It is often called a sea scorpion, though it was not a true scorpion, but rather a large predatory arthropod that lived in water. This animal existed during the Early Devonian period, around 395 to 390 million years ago, and became extinct toward the end of the Devonian. Jaekelopterus Rhenaniae was one of the largest aquatic arthropods of its era. Its body length is estimated at about 2.3 to 2.6 meters, making it far larger than a human. The name Jaekelopterus honors Otto Jaekel, while Rhenaniae refers to the Rhineland region in Germany, where the fossils were found. One of the most important discoveries was made in the Clare Formation in Germany, where a huge 46-centimeter-long claw was found. Based on this fossil, scientists calculated the animal's full body size. Its exact weight is unknown because only partial remains have been preserved, but it was undoubtedly very large and heavy. The name was first given in 1914 by the German paleontologist Otto Jaekel, who described it as Pteron Rhenaniae. Later, after further research, it was classified into a separate genus and renamed Jaekelopterus. In 2007, important remains were discovered in Germany by Markus Poschmann, Simon Brady, and Eric Tetley, which provided new insights into the size and biology of this creature. Jaekelopterus Rhenaniae mainly lived in freshwater habitats, such as rivers and swamps. It was a predator that fed on smaller fish, aquatic arthropods, trilobites, and other creatures. The animal also had large compound eyes adapted for vision underwater. Its large clawed appendages were used to grasp and tear apart prey. Its body was flat, segmented, and covered with a hard exoskeleton, while its broad tail section helped with stability in swimming. The extinction of Jaekelopterus Rhenaniae was likely caused by several factors. During the Devonian period, changes in climate, the drying of water habitats, increased competition from other predators, and the decline of available food resources contributed to its disappearance. Today, there are no direct descendants of this species. It was not the ancestor of modern scorpions or spiders, but rather a side branch of chelicerate evolution that eventually died out. For this reason, it is known only through fossils, but in its time, it was one of the most impressive and formidable predators of its environment. In general appearance, Jaekelopterus resembles other pterygotid eurypterids, with a large, expanded telson, the rearmost body segment, as well as enlarged claws and forelimbs. Both species of Jaekelopterus were initially described as species of closely related genera, but were later placed in a separate genus based on differences observed in the genital appendage. Although this feature was later shown to have been misidentified, other distinguishing characteristics separating the genus from its relatives were identified, including a triangular-shaped telson and differently inclined teeth on the claws. The chelicerae and compound eyes of Jaekelopterus indicate that it was an active predator with strong visual acuity, likely serving as the apex predator of the early Devonian Euramerican ecosystems. Although eurypterids such as Jaekelopterus are often referred to as sea scorpions, the strata in which its fossils were found indicate that Jaekelopterus lived in freshwater environments. Jaekelopterus is the largest known eurypterid and the largest arthropod to have ever existed. It is known from a chelicera, claw, measuring 36.4 centimeters, 14.3 inches long, but with a missing quarter of its length, meaning the full chelicera would have measured 45.5 centimeters, 17.9 inches, discovered in the Emsian Clare Formation near Wilwerath, Germany. If the ratio between chelicera length and body length was similar to that of other giant pterygodids, such as Acuteromus and Pterygotus, the animal's estimated total body length would have been between 233 and 259 centimeters, 7.64 to 8.5 feet. When the chelicerae were extended, the total length would increase by another meter. According to these estimates, even without the extended claws included, Jaekelopterus would exceed the maximum body size of all other known giant arthropods by almost half a meter. Morphologically, Jaekelopterus resembles other pterygotid eurypterids, but is distinguished by its triangular telson, the last body segment, and the curved main denticles, teeth, on the movable part of its claws, cheliceral rami. Pterygodids, an advanced group of eurypterids, differ from other families in several ways, especially in their chelicera and telson. The chelicerae of Pterygotidae are large and robust, clearly adapted for active predation, and their chelae, pincers, resemble those of some modern crustaceans, with well-developed teeth compared to other eurypterid groups. Another distinctive feature of the family is their flattened and expanded telson, which likely functioned as a rudder during swimming. Known from more than 30 specimens, howelli shows an almost identical denticulation pattern in the chelicera as Rhenaniae, and it also retains a flattened posterior margin of the telson, which is triangular in shape, as in Rhenaniae. However, it differs clearly from Rhenaniae in having a serrated telson margin and a massively elongated second intermediate denticle. In addition, the type A genital appendage is not divided at the tip. Howelli is significantly smaller than Rhenaniae, with a total length of about 80 centimeters, 2.6 feet. Jaekelopterus was first described as Pterygotus Rhenaniae in 1914 by German paleontologist Otto Jaekel, based on an isolated pretelson from the Lower Devonian of Aachen, Germany. Jaekel considered the pretelson to be characteristic of Pterygotus, as other elements found did not differ much from previously known species of that genus, such as P. buffaloensis, and he estimated the living animal to be about 1 meter long, or 1.5 meters, including the chelicera, 3.3 to 4.9 feet. Norwegian paleontologist Leif Sturmer later provided a detailed description of the species in 1936, based on more comprehensive material discovered near Overath, Germany, by German paleontologist Walter Gross, including parts of the genital appendage, chelicerae, the metastoma, a large plate forming part of the abdomen, and telson fragments. Sturmer interpreted the genital appendage as segmented, distinguishing it from other Pterygotus species. In 1964, British paleontologist Charles Waterston established the genus Jaekelopterus to accommodate Pterygotus Rhenaniae, arguing that it differed sufficiently from other Pterygotus species to warrant its own genus, mainly due to differences in the genital appendages. Waterston diagnosed Jaekelopterus as a pterygotid with segmented genital appendages, a trapezoidal prosoma, narrow and elongated chelicerae with terminal teeth set at nearly right angles, and primary teeth that are slightly forward-angled, along with a telson having an expanded spine and posterior region. The generic name honors Otto Jaekel. The Greek word pteron, meaning wing, is a common epithet in eurypterid nomenclature. In 1974, Sturmer created a new family, Jaekelopteridae, to accommodate Jaekelopterus, based on the presumed significant differences in genital appendages between it and other pterygodids. However, this distinguishing feature was later shown to be based on a misinterpretation from Sturmer's 1936 study. The genital appendage of Jaekelopterus was actually not divided and resembled that of Pterygotus. As a result, the family Jaekelopteridae was later rejected and regarded as a synonym of Pterygodidae. Another species of Pterygotus, P. howelli, was named in 1952 by American paleontologist Eric Kellisvig Waring and Stormer, based on fossils of a telson and tergite, the dorsal part of a body segment, from the Lower Devonian Beartooth Butte Formation. The species name honors Dr. Benjamin Howell of Princeton University, who had loaned photographic samples for study to Kjellesvig Waring and Sturmer. In 2007, Norwegian paleontologist Erik Tetley reassigned the species to Jaekelopterus as Jaekelopterus howelli. Jaekelopterus belongs to the family Pterygotidae within the superfamily Pterygotioidea. It closely resembles Pterygotus, differing mainly in the structure of the genital appendage and possibly some telson features. The close similarity between the two genera has led some researchers to question whether the pterygotids are overdivided at the generic level. Based on similarities in genital appendages, American paleontologists James Lamsdell and David Legg proposed in 2010 that Jaekelopterus, Pterygotus, and even Acutioramus may be synonyms of one another. Although differences have been noted in the chelicerae, Waterston, 1964, questioned the reliability of such traits for distinguishing eurypterids, since their morphology is closely tied to lifestyle and can vary throughout ontogeny, post-embryonic development. While telson morphology can distinguish genera among eurypterids, Lamsdell and Legg noted that the triangular telson of Jaekelopterus could still fall within the morphological range of the paddle-shaped telsons found in Pterygotus and Acutioramus. Even genital appendages can vary within a genus. For example, in Acutioramus, they change from spoon-shaped in early species to bilobed forms, eventually resembling that of Jaekelopterus. Lamsdell and Legg concluded that to determine whether these genera are true synonyms, an inclusive phylogenetic analysis involving multiple species of Acutioramus, Pterygotus, and Jaekelopterus would be required. The following cladogram is based on 9 of the most well-known pterygotid species and 2 outgroup taxa, Slamonia acuminata and Humillaria socialis. Since Jaekelopterus was described as a separate genus by Waterston in 1964, it has been classified as the basal sister taxon to all other species within Pterygotidae, because it was thought to possess segmented genital appendages, which in other pterygotids are fused and undivided. However, re-examination of the specimens later determined that these were misidentified genital appendages. Pterygotid eurypterids include the largest known eurypterids, such as Pterygotus and Acutioramus. Several factors may have contributed to the exceptional size reached by Jaekelopterus, its relatives, and other Paleozoic invertebrates, including predatory behavior, mating competition, ecological rivalry, and environmental resource availability. However, factors such as respiration, energy expenditure for movement, and the physical limitations of the exoskeleton restrict the size of arthropods. Aside from their robust and well-sclerotized claws, most preserved body segments of pterygotids are thin and poorly mineralized. Even their tergites and sternites, the plates forming the dorsal and ventral surfaces of abdominal segments, are usually preserved as paper-thin compressed structures, indicating that pterygotids had a very lightweight build. Similar lightweight adaptations are seen in other Paleozoic giant arthropods, such as the millipede-like Arthropleura, and have been suggested to be crucial for the evolution of arthropod gigantism. A lightweight body design would have reduced the impact of size-limiting physiological constraints. Despite being the largest arthropods, the light body structure of Jaekelopterus and other large pterygotid eurypterids means they were not necessarily the heaviest. Other giant eurypterids, particularly the deep-bodied, walking forms of the family Hibbertopteridae, such as Hibbertopterus, which reached lengths of nearly 2 meters, may have rivaled or even exceeded the pterygotids and other giant arthropods in mass. In 2008, American paleontologist Alexander Kaiser and South African paleontologist Jaco Klok argued that the size of Jaekelopterus had been overestimated, noting that the ratio between chelicera length and body length would not necessarily remain constant as the animal matured. The teeth on the claws showed positive allometry, being proportionally larger in larger specimens, and Kaiser and Klock suggested that this might apply to the entire Chelicera. Furthermore, the largest known coxa, leg segment, of the species, measuring 27 centimeters, 11 inches wide, would correspond to a total body length of only about 180 centimeters, 5.9 feet. Positive allometry has not been observed in other eurypterid genera, including close relatives of Jaekelopterus, and is not generally found in eurypterid chelicerae. Some undescribed specimens of Jaekelopterus Rhenaniae feature large chelicera-like appendages, including another claw found in the same strata as the original specimen. In response that same year, Brady, Poshman, and Tetley maintained that the previously estimated maximum size of around 2.5 meters, 8.2 feet, remains the most accurate estimate for the species, true length. Like all arthropods, eurypterids grew by passing through a series of molting stages known as instars. Unlike many other arthropods such as insects and crustaceans, chelicerates, including eurypterids like Jaekelopterus, horseshoe crabs, sea spiders, and arachnids, are direct developers, meaning they do not undergo drastic metamorphosis after hatching. Modern xiphocerans, horseshoe crabs, hatch without fully developed opisthosomal appendages attached to the posterior body segments, whereas modern spiders are complete direct developers. Studies of fossil specimens of Strobilopterus and Jaekelopterus indicate that eurypterid ontogeny paralleled that of modern horseshoe crabs, but eurypterids, like arachnids, were true direct developers, hatching with the same number of appendages and segments as adults. Although several fossil specimens of Jaekelopterus howelli are known, their fragmentary and incomplete condition makes it difficult to study the species' ontogeny in detail. Nevertheless, significant changes occur in the chelicerae, telson, and metastoma as the animal matures. Among the howelli specimens examined by Lamsdell and Selden, 2013, 4 preserved the chelicerae well enough to study the denticles, tooth-like projections. 2 of these chelicerae were thought to belong to juvenile individuals, and the other 2 to adults. The overall cheliceral morphology was similar across all ages, with the same number and arrangement of denticles, though notable differences were present. Specifically, the primary teeth became progressively larger relative to the intermediate ones. In juveniles, they were about 1.5 times larger, whereas in adults, they were 3.5 times larger. Additionally, the terminal denticle in adult specimens was much larger and more robust than in juveniles. Most strikingly, the second intermediate tooth, which was no different in size from the other intermediate denticles in juveniles, became massively elongated in adults, reaching twice the length of any primary tooth. While similar patterns of tooth growth have been reported in other pterygoid genera, this dramatic ontogenetic elongation of the second intermediate denticle appears to be unique to Jaekelopterus, particularly to J. howelli. The metastoma of Jaekelopterus also changed in proportion as the animal matured. In J. Rhenaniae, the relative width of the metastoma decreases through ontogeny. Similarly, in J. howelli, the metastoma is broader in juveniles than in adults, although the difference in length-to-width ratios is minimal, approximately 1.43 in juveniles versus 1.46 in adults. Comparable changes in metastomal proportions have also been documented in other eurypterid genera, such as Stormeropterus, Moselopterus, and Strobilopterus. The cheliceral morphology and visual acuity of pterygoid eurypterids distinguish them into specific ecological categories. The main method of determining visual acuity in arthropods involves measuring both the number of lenses in the compound eyes and the interomotidial angle, IOA, the angle between the optical axes of adjacent lenses. The IOA is especially important because it can be used to infer ecological roles, being lower in modern predatory arthropods. Jaekelopterus Rhenaniae and Pterygotus anglicus had high visual acuity, as indicated by the large number of lenses in their compound eyes and the low IOA values. Further studies of Rhenaniae's fossilized eyes, including a large specimen preserving the right eye from the Upper Sigenian, as well as smaller, likely juvenile specimens, confirmed the genus's high level of visual precision. The average IOA for Jaekelopterus, 0.87 degrees, is comparable to that of modern predatory arthropods. Visual acuity increased with age in Jaekelopterus. Smaller specimens had poorer vision. This pattern is consistent with that seen in other pterygotids, such as Pteron, suggesting that adult Jaekelopterus likely inhabited darker environments, such as deeper waters. Fossilized eurypterid trackways support this interpretation, showing that eurypterids likely migrated to shallow, nearshore habitats for mating and reproduction. Jaekelopterus also had overlapping forward fields of vision, implying stereoscopic vision, characteristic of active predators. Structurally, eurypterid eyes were nearly identical to those of modern horseshoe crabs. The square-shaped receptor cells in the compound eyes of Jaekelopterus resembled, though did not exactly match, those of horseshoe crabs, suggesting a specialized visual system. The photoreceptor cells in Jaekelopterus were remarkably large, about 70 millimeters, compared to 1 to 2 millimeters in humans and most other arthropods, but similar in size to those of modern horseshoe crabs. The unique eyes of modern horseshoe crabs differ greatly from those of other living arthropods, providing enhanced peripheral perception and contrast detection under low and scattered light conditions, abilities crucial for animals living in dim environments. Given the strong similarities, Jaekelopterus likely possessed comparable adaptations. Its highly specialized eyes made Jaekelopterus exceptionally well-suited to a predatory life. The morphology and body structure of Jaekelopterus and other eurypterids within the family Pterygotidae indicate that they were fully adapted to an aquatic lifestyle. According to a 2007 study by Brady, Poshman, and Tetley, it is highly unlikely that arthropods of the size and build of Jaekelopterus could have walked on land. Although eurypterids like Jaekelopterus are commonly referred to as sea scorpions, the deposits in which Jaekelopterus fossils have been discovered suggest that it lived in non-marine aquatic environments. The Beartooth Butte Formation in Wyoming, where J. howelli fossils were found, has been interpreted as a calm, shallow river environment. This species was discovered alongside two other eurypterids, Dorfopterus angusticalis and Strobilopterus princetonii. Similarly, fossil remains of J. Rhenaniae from Rhineland have been interpreted as part of a shallow water habitat that ranged from brackish to freshwater conditions. The chelicerae of Jaekelopterus were large and robust, featuring curved movable rami and denticles of varying lengths and sizes. Some puncture wounds observed on fossils of the Devonian agnonfish Lecreaspis patula from Utah may have been caused by Jaekelopterus howelli. Recent studies indicate that Jaekelopterus was an active and visually oriented predator. Fully grown Jaekelopterus individuals were likely apex predators in their environments, preying on smaller arthropods, possibly including members of their own species through cannibalism, as well as early vertebrates. As a powerful and agile predator, Jaekelopterus was probably capable of swift and precise movements. The hydromechanics of the swimming paddles and telson of Jaekelopterus and other pterygodids suggest that all members of this group were capable of swimming, forward propulsion, and rapid turning maneuvers. Although they may not have been exceptionally fast swimmers, they were likely capable of ambushing prey effectively within habitats such as lagoons and estuaries.